To introduce our next speaker, I'm going to first welcome Socrates Trujillo to the podium. Socrates reached out about a month ago and floated an idea that he and Marianne are going to discuss a little bit more in this next presentation. And it really does get further upstream in the knowledge, in the knowledge trajectory, and that is looking at what are some of the opportunities for further research. So we've talked already a lot about some of the practice, the gaps in practice, and the gaps in educational delivery, and the gaps in inspectional or regulatory activities. And so this is going a little bit further upstream and saying, okay, so what can we do to generate new knowledge, new appropriate knowledge that will help address some of this? So with that, Socrates.
Thank you, Chris. Hi, everybody. I'm Socrates. Who you don't know me, I used to be the Producer Safety Network representative from CIFSEN here in the Northeast. And when I got offered the opportunity to work with this incredible team of researchers at the same center but a different office, the only thing I wasn't sure was how I could come back to continue working with you all. So fortunately enough, my supervisor, Marianne Solomotis, and also the director of the director of research for the office that I belong to, liked the idea to come and explore anything that we could do with you. So the intention that we have here is just to give you a little brief overview of what we do at the Office of Applied Research and Safety Assessment at CIFSEN, and what we want to hear from you. And please help me welcome Dr. Marianne Solomotis. Thank you.
Thanks, Socrates. Can everybody hear me? I'm always told I should project my voice more, but anyway, thank you. First, thank you to Chris and Elizabeth for squeezing us in this really ambitious agenda. We really appreciate it. Especially for me, this has been a great opportunity and a great experience this morning. As the name of our office says, we are a regulatory research office. So that's what we do, is research. Regulatory is Samir and his folks. So we do the research, and we provide the information to them, and they do what they want with the information. So besides wanting to share what we're doing, and as Socrates said, maybe identify some potential collaborations, we also want here to identify additional data gaps that we can work with together and also leverage our resources. Before I start, I just want to briefly acknowledge the people who actually do the work. First, my supervisor and office director, Mary Torrance, who basically established the Produce Safety Research Consortium. We always joke that she's the mother who gave birth to the baby, and I'm the nanny who's nurturing it. So I don't know how good a job I'm doing, but I try. And then Socrates, who's been with us for 2 months and has already proved himself a great asset. I mean, he's always a step ahead of me. Every time I ask him something, he's already done it. And then there's our bacteria, metagenomics, soil chemistry people, our virology people, cyclospora, Josie Freeman, who takes care of our samples, and Elizabeth Denard, who keeps me on my toes and helps me with the coordination, scheduling meetings, ensure that everybody's where they're supposed to be. So basically, you know about FSMA rules and what it's all about more than I do. I just want to reemphasize where some of our data gaps are and where we need to do what we think we need to do additional research is ag water, biological soil amendments, the worker health and hygiene, equipment tools, everything that's basically been already presented and mentioned here. Okay, so let's talk about the ORSA Produce Safety Research Consortium. As I said, Mary Torrance established this, and it's made up so far of 3 universities: NC State University, the Ohio State, UGA, and USDA or ARS at the Salinas Valley in California. And the goal was to have different people in different areas so we can do different regional comparisons. So, and it was to leverage ongoing projects, facilitate collaborations, provide produce-related on-farm environmental collections, which otherwise, as a regulatory agency, we wouldn't be able to get for research purposes, facilitate and fill data gaps, and expand our contributions to complement the other office what I didn't mention is also is one of 2 research officers at CIFSAN. So they've already been doing produce safety for several years. I'm sure you all know them, the Office of Regulatory Science. And we're just complementing what they're doing, trying not to duplicate anything. Okay, let's go try this again. Oh, okay, thank you. So the specific objectives were basically to compare the prevalence of different bacterial pathogens. Salmonella, we focused on Salmonella, Listeria, Campylobacter, Aztec. And what we're doing, a lot of people aren't doing, is looking for viruses in the environment and Cyclospora. We want to compare, as I said, regional and farming practice differences for the soil microbiome and the presence of pathogens. We were asked to focus on raw manure, treated manure, and untreated manure. And we want to investigate the presence of pathogens and their survival in different water sources and different irrigation types. And as I said, drip tape where applicable. For example, the Amish farmers in Ohio use a lot of drip irrigation. And at the end of the season, they have the drip tape. They send us samples, and we can look at the microbiome and what's in that drip tape. Again, we're also interested in the physical and chemical properties and moisture content of the soil and how that will impact pathogen survival, and look at the relationships between the pathogens and the normal microbiome. Again, we're looking at the microbiome for potential indicators of pathogens and also for competitors, and that will help us develop methods that will enrich for pathogens. And we're looking at the microbiome soil, water, produce. We're looking for presence of antibiotic resistance genes. And this year we will start looking at taking air samples as well. And one of the main things one thing I'm really interested in, and I know the Division of Produce Safety is looking at the potential transfer of the pathogen to the plant and eventually to the produce. And of course, we need metadata. We need to look at the impact of different amendments. And animal husbandry, do the farms have animals on the farm? Like one of the farms we know actually get calves and they grow them up and they take care of them until they're adults and then give them back to the animal farmers. And then we look at also the CAFO, the distance of animal feeding to the planting fields. So how does the Produce Safety Research Consortium help us? Well, they provide us with blind samples, water, soil, produce from the natural environment, and they're all blind. We know the area or the region of the farms. We have no idea which farm the samples come from. They also do some isolations for us and provide us with DNA. And they also are a conduit for us, between us and the farmers. And what goes on in the farms, because we can talk to the farmers ourselves. So basically, it's just pieces of the puzzle. How do we get from the air, the impact of the air, the water, the soil, the amendments, irrigation, to what's in the end product? And this, I'm going to go, there's a whole lot of different amendments, and all of them impact the microbiome differently. And this is just, it's next generation sequencing, which shows everything that's in the sample. And as you can see, there's a significant difference between, on the left-hand side, field soil and composted soil. So again, you know there's something going on there that may or may not be an enrichment for a pathogen. We've also looked at 3-month intervals of the same soil, and there's some separation, some change, but on the whole, it's not as significant as the different soils. Okay, we've also doing we have Susan Leonard, who's doing some collaborative work with Maria Brandl from the Salinas Valley, USDA, and we're looking at the impact of modified atmosphere packaging on the survival of E. coli 0157. So basically, they harvest the lettuce, they cut and wash it, they inoculate it with 0157, and then put it in modified atmosphere, and they send it to us. So this is what a metagenomic graph or next-gen sequencing, and again, you can just see everything that's in that lattice. And I'd just like to, I don't know where, I'm not even going to try that, but if you look at the blue, at all the columns, before I continue, we have two types of lattice, which is the DA, which has a good shelf life, and the TT, which has poor shelf life. And if you look at the graph from the unprocessed lattice, you can see those blue rectangles. Those are all Pseudomonas species. And you can see there's a difference between the good shelf life lattice and the bad already. After storage at 6 degrees and refrigerated, you can see a huge difference in the trend of how the Pseudomonas species have decreased. And then you see those orange or peachy colors, that's Owenia and Enterobacteriaceae. And you see how they've actually come up and they weren't present before. So that, and we also have E. coli 0157 seems to grow and survive in the TT after storage. And we can't see them there because the relative abundance is not sufficient, still pretty low, to be visible in the graph. But again, this is just another experiment that shows, or study that shows, depending on the lattice type, what kind of growth you can get in survival pathogenicity. So in summary, this is our third year. We're doing this work with the consortium. We have preliminary data, as we haven't not analyzed all our data yet. We're still in the process of testing, and this is only going to end at the end of September. One of the things we found is the soil between Georgia and Ohio is very different, both in moisture and in texture and in the soil chemistry. The one thing that's really exciting about this whole Produce Research Consortium is, I'm sure you all know FDA has the BAM methods. Well, the BAM does not work for soil, does not work for water. In fact, one of the prime examples of the viruses, our virologist Effie Papafranco, she's been trying to develop a method for detecting viruses in soil. And she finally got this great method. It worked, she repeated it, somebody else repeated it. Then we get an amended soil, nothing. The inhibitors were completely unbelievable. So we're back to square one. So it's really helping us develop methods as well, and we're finding a new role for metagenomics. So our future directions, the end of this year, once we have all the data, we want to analyze the 3-year data to determine where to focus, and then we can go on and on and on, surveying the land, but we are finding some trends and patterns, and we'll see where we should focus, maybe on specific farms, maybe on something else, on specific characteristics. And the exciting thing for me is we're establishing, as we speak, what we called the also Produce Research Facility, and we actually have 6 small growth chambers on order. We have 3 rooms which are going to change into huge growth chambers. And now we're going to try and simulate what goes on in the environment with these growth chambers. And this is where we will request, or we'll hope we'll get some collaboration from people who have experience. And Socrates who's basically in charge of this establishment, has a lot of other good ideas. So thank you for your attention. And again, we wanted to do this to identify potential collaborations, leverage resources, and identify data gaps. Thank you.
Thank you very much. Any follow-up questions? Don?
Yeah, so is the lettuce microbiome stuff published yet, or is it going to be submitted soon?
No, it's not published. We have a few samples left to sequence. Oh, sorry, thank you. No, we're finishing that off. We have a few samples to sequence before we finish it off.
And are you going to present it maybe at IFP this summer?
In fact, Maria Brandl presented that last year at IFP.
Okay.
I'm not sure if Susan will be presenting it at IFP this year.
Thanks.
Other questions? Okay, I suspect all of the thank you both very much. I suspect all the conversations we've already had in this afternoon session will be continued in the next activity we're about to do.